Students, in today's class, we'll start our discussion with seed production. Am I audible to you all? Just give me a second. So you all know, like today the application has been released. So students are already started asking me like when will be the exam, like tentative date that exam will be conducted. So exam can be in somewhere around July end. Okay, within July end, you will get your exam. So June 16 is the last date for applying. I think like it is the last date. So after that, it is another 10 to 15 days that they will announce the exam. And after that announcement of the exam date, it will be around 1 week or 2 weeks of time. So this is the ideal case, and there is also a possibility that it can still go late. But in any case, like when they release the application, it's around 10 to 15 days that they will release the date. So once they are releasing the date, it is again for 1 or 2 weeks that you will get your exam. So it might be tentatively on around July 2nd, 3rd, or 4th week, not first week, July 4th. Because see, if they are closing the application by June 16, so another might be another uh, 1 week they might release the exam date. So once they release the exam date, they will at least give you 15 days of time from the date of announcement of the exam to the conduct of exam. So your exam might be conducted somewhere between 15th of July to 30th of July. Okay, so it might still go late. There are possibilities, but since they have already released the application, believe like they are not making a delay in it. They will immediately release the exam date soon after you just apply this application. Soon after the student submit the application, last date is over. Might be after 1 week they will release the date of the examination. Yeah, 15 to 20 days after closing the application, they will give a exam date. Again, once they release the exam date, it is again 1 or 2 weeks that the time will be there for the date of examination to start. Okay, so my screen is visible to you all. Yes, like you all are able to see my screen, the seed production screen. Okay, fine, thank you. So in today's class, we are going to start our discussion with seed production. So I am only left out with another two topics apart from the seed production, which is seed processing and seed testing. So let's see like how many classes that it will take. I want to cover them in a single class, like one class for seed processing and one class for seed testing. But I don't know like how many days it will take. It might take 1 or 2 classes to finish each topic. So hardly by this weekend we can finish the syllabus. That is for sure. So before going to the topic, this is a today's content outline where we will be discussing about the mode of pollination and the factors for variety deterioration and SMR and SRR. Yes, SMR and SRR, which is seed multiplication ratio and seed replacement ratio, and the methods of hybrid seed production, and the different schemes for seed production that we have in India, and the area for seed production. What is the ideal area that is required for the seed production? And then we will discuss IMSES. It's Indian Minimum Seed Standards, seed certification standards for seed production. So I already told you that there is a one file in a AgriEdit app in a free material section where you can go, where everyone can go and download it, in which I have mentioned the isolation distance, designated diseases, in which I will also include the planting ratio and SMR. So these two are not included yet, but I will do it as soon as possible. for now it has isolation distance and designated diseases associated with the particular crop. Okay, so just go through the document once I upload the rest of the content, I will update you in the group. So this is the seventh part, and lastly we will discuss the seed production of different crops. So the different crops, we have a vast amount, like vast round of crops that are need to be discussed. Like if we include the field and horticulture crops, it will take at somewhere around 100 crops that are there. So out of 100 which we scale down, we can discuss around 10 to 15 of them, which are very important, in which we'll cover the cereals, pulses, oil seeds, fiber crops, and vegetable crops. So we'll try to cover them. Anyhow, like they are self-explanatory. I've just considered, like I have just collected all the information that is very much required, and I will tell you like what are the important things that are to be covered in these particular seed production aspects. So you just concentrate on them and just try to revise them from time to time. So rest of the things are like you can understand everything that I've just mentioned in these notes. So let's see how it goes. Starting with mode of pollination, like this is again a overlapping content from the breeding, where you have already known that there are two different types of pollination behavior, which is of self and cross. So what are the mechanisms that favors the self pollination? If you remember, just directly show you the things, that would be better. So the self pollination is nothing but the autogamy, which is the closest form of inbreeding, also called as selfing, and then it is cross pollination, which is also called as allogamy. So the mechanisms that favors the autogamy or the self-pollinations are bisexuality, homogamy, cleistogamy, chasmogamy, and the positions of the anthers that are very closely surrounded with the stigmas, or stigmas are surrounded by the anther itself, so that there will be a self-pollination and there won't be any crossing. And next it is a mechanism favoring allogamy. In the autogamy, so this particular autogamy will lead to the homozygosity. I hope like you people know it. And such species which develop the homozygous balance, they literally do not exhibit inbreeding depression. So this is what you should understand whenever the certain crop species is enjoying the homozygous nature or the homozygous balance, it is evident that it won't be suffering from the inbreeding depression. So the case of inbreeding depression we don't see in the self-pollination. So when we talk of allogamy, this is completely different or opposite, where it is produced by the cross-pollination, and this is a common form of outbreeding. And this allogamy will leads to the heterozygosity in the heterozygosity for the alleles. So once it is developing the heterozygosity or leading to the heterozygosity, it will develop a heterozygous balance, or it is enjoying the heterozygous balance. That is the reason when it is treated or when it is continuously selfed, so it will definitely exhibit the inbreeding depression. On the continuous selfing for 6 to 7 generation, you will see the inbreeding depression in the cross-pollinated crops. So what are the different mechanisms that favors the cross-pollination? So before that, just try to remember the examples of all these things, like position of the anthers, chasmogamy, cleistogamy, homogamy, and bisexuality. So they are very important. Next, it is about dichloeny. Dichloeny is nothing but having the unisexual flowers, in which they are divided into monoecious and dioecious. And next we have dichogamy, which refers to the maturation of the anthers and the stigma at the different times. So there are again two different types, which is protandry and protogeny. Protandry, where the anthers matures first, and the protogeny, the pistil matures first before the anthers. So there are other examples. Just add the examples wherever you see in the books. So just take a printout, or you can just add it in the same file. So make sure to remember as much as examples that you can for this protandry and protogeny. Next we have heterostyle, where the style and the filaments in the flower are of different length. This particular example is typical for linseed. So next we have archogamy. This is just an endurance of the self-pollination because of presence of some barriers, which are physical barriers. So this is particularly seen in the case of alfalfa. So what is the mechanisms that will break or that will allow the cross-pollination to occur in the alfalfa, even though these physical barriers are present? How the pollen is excised? Screen is not visible. Is this same for everyone? Because I am able to see my screen. was, just please, anyone confirm me, like others? Okay. Mustaida, it might be because that unstable internet connection, it will restore. Just wait for some time. Okay. I just ask you one question, which was regarding the alpha alpha. Just can you name the mechanism which will break this anther and bust out the pollens out of it so that there will be a pollination, or more specifically, the cross pollination is because of this particular mechanism in case of alpha alpha. Alpha alpha, on the what is the other name? Yes, it is tripping. There is a tripping mechanism in case of alpha alpha where this barrier is broken down, and by which, and this barrier is broken down by the insect activity. So this is how it leads to the cross pollination. So next we have also have certain other mechanisms which are self incompatibility and male sterility, which will prevent this self pollination. So what is the difference between incompatibility and sterility? Incompatibility means the fertile pollens will fail to fertilize the same flower. So it is an inability of the fertile pollen to fertilize the same flower, but it can fertilize the different flower of the different plant. So this is the case of self incompatibility, and this is more commonly seen in case of brassica, radish, nicotiana, and many grasses, grass species. So next we have male sterility, and this is also seen in the cole crops. Okay, self incompatibility, and this is used in hybrid seed production for the commercial purpose. So next it is about the male sterility. Male sterility is nothing but the pollen grains themselves are non-functional, whether it is self-pollination or cross-pollination, or whether the pollens are landing on the same flower or a different flower on a same plant or a different plant. They are not functional at all. So those type of that type of mechanism is because of the male sterility. Clear? So we have other two terms, which is geitonogamy and xenogamy. So what is geitonogamy? So geitonogamy is nothing but the transfer of the pollen from the anther to the stigma of a different flower. Now the pollen transfer is actually taking place from the anther to the stigma of the different flower, but that another flower or the stigma is present in the same plant. For example, this is a particular plant, and above there are the pistil, and below there are the anthers, or above there are the anthers and below the pistil. So this anther, the pollen will fall on the stigma of the pistil or the flower, which is present below this, but this is again within the same plant. So in this case, it is called as geitonogamy. So one typical example is, this is in case of maize. So since it is occurring in the same plant, this is again the genetically a self-pollination. Though we call it as a cross-pollination because the transfer of the pollen is taking place from the anther to the stigma of a other flower, but it is genetically the self-pollination only. Okay. So there is another term called as xenogamy. So who will tell you the meaning of xenogamy now? Yes, what do you understand by xenogamy? So this is xenogamy is just the opposite of geitonogamy, where the anther transfer or the pollen transfer from the anther takes place to the different or to the stigma of the different flower, which belongs to the different plant, or more easily it can be said as cross pollination. Xenogamy is a mechanism of cross pollination where the transfer of the pollen from the anther to the stigma of a different plant. I hope like you are understanding the terms, meaning of all these terms, because if in the exams you might expect the direct questions that might confuse you. So make sure to understand the meaning of all these terms. Okay. So next, I have just listed out the characteristics of the wind pollinated crops. What are the typical characteristics that the plants have in order to facilitate the wind pollination by wind, and also like what are the characters that the plants possess in order to favor the self pollination. Just go through all these points so that if anything is asked in the exam, you'll be able to answer them. So I have just more clearly mentioned about all the adaptations that are favoring the cross pollination. So first adaptation is for wind pollination, and the second adaptation for insect pollination. There are different plant parts which will favor either the wind pollination or the insect pollination. So those floral adaptations you need to take care of. Okay. Nextly, we can discuss about the deterioration of the crop varieties. So can you just tell me like what are the common reasons for the deterioration of the variety? So for example, if the variety is in the public domain for around 8 to 10 years, so we normally say that a variety lifespan is around only 5 to 6 years. So after that, the variety will deteriorate. So what is the main reason for this deterioration? Or the breeders themselves have like incorporated certain genes in them that they will deteriorate after some time? Or is there any other mechanisms by which the deterioration is taking place? Or any natural phenomena that is occurring by which the variety deterioration proceeds from year to year? So what do you think? Just asking about the deterioration of the varieties. Yes, one reason might be the mutations. Very good. Natural outcrossing. Natural outcrossing, varietal mixture. Yes, varietal mixtures can also be called as mechanical mixture. Yes, admixtures. Very good. So there are certain reasons for the deterioration of the variety. So they are the developmental variation, the variations that takes place because of the varied environmental conditions that pass from generation to generation. They are called as developmental variations. So this should be like developmental variations. Next one is mechanical mixtures. So during the harvesting, like this mainly occurs during the harvesting time, where there are the cases of mechanical mixtures by which the variety will be deteriorated from generation to generation. Next, it is because of mutations. Certain stable morphological or certain economically important characters will deteriorate, or it will change because of the mutation in the genes responsible for that particular trait. So that is again the mutation. And next, because of natural outcrossing, so there will be change in the allelic frequency by which the complete expression of the variety will be changed. And then genetic drift, we see the genetic drift where there will be loss of alleles. Next, because of some minor genetic variation that we observe in the nature because of again the varied environmental conditions. And seventh point, it is because of the influence of the diseases. Whenever there is a strong influence or the attack of a diseases or the infection is taking place, it will lead to the massive loss in the yield and everything. So from generation to generation, the ability to tolerate this stress will be decreasing. So this is what deterioration means, right? So next, the techniques that are used by the breeder in the seed production will also lead to the deterioration. So if the genetic purity is not maintained from generation to generation, eventually the variety that was developed at the first place for some economical like economic character, that will be eventually deteriorated. Why? Because they are not maintaining the genetic purity. Genetic purity in the sense like the genome constitution. If the genome constitution is not exactly the same of how it or when it was developed, if it is not same after certain generation, then obviously the characters that it is expressing will be different, right? So this is how it is the reason. This is also a reason for the variety deterioration. And whenever there is a breakdown of the male sterility in the hybrid seed production, so this deterioration will also occur. If they have developed certain variety which is a male sterile line, which is always used as a male sterile line or a female line, so from due to the breakdown of this male sterility, it cannot be used as a female parent. So this will also lead to the deterioration. And lastly, it is improper or defective seed certification system. So why do you think like the concept of certification came here? See, the seed multiplication in India or the production of quality seeds in India is mainly regulated by certification system. So why the quality regulation is very important, and what are the attributes of the quality seeds? Again, they are genetic purity, physical purity, germination, moisture content, free from pests and diseases. So all these are the standard parameters. So like any loss in the parameters or any decrease in the percentage of these parameters will have a direct effect on the variety. So that is again nothing but the deterioration. That is why the improper certification system will be also the reason for the deterioration in the variety. So since we are talking about the seed production, I thought this is important for now to be discussed. That is why I've included this. Okay. So what are the different ways to prevent them? So how can we prevent them? So now we came to know about the problems and different sources of how the deterioration can occur. So what are the different mechanisms that will prevent this contamination? So first thing is, at the very first place, you need to control the seed source from where it is coming in the multiplication chain. And next, the preceding crop requirement. Whenever you sow the seed in the field, you just check the preceding crop requirement. What are the crops that have been cultivated in the preceding generations, so that there won't be any mechanical mixtures or any loss to the genetic purity in the upcoming generations or upcoming season. And next is maintaining the proper isolation distance will help in the production of quality seeds, as we have understood it already. And thorough roguing. So what is the effect of thorough roguing? Roguing will remove the off types from the fields, including the weed seeds, weed seeds which are objectional, the seeds which are of other crop plants, which are off types of a similar variety. So since the roguing plays a very major role, it will help in maintaining the quality of the seeds. Next is, yes, stringent seed certification procedures will help to prevent the contamination and the grower test. So this can be again included in the seed certification, where we do the grower test in order to check the genetic purity of the variety. So by the thorough checking of this genetic purity from for generation to generation, whenever the seed production is taking place, we can prevent the contamination. Anyhow, the deterioration will take place eventually, but premature deterioration or early deterioration can be prevented by these methods. So the life validity of any variety cannot be more than 8 to 9 years, because there are certain things that are out of control of the humans, like that humans cannot control. So it's around, like on an average it is 8 to 10 years you can take it. So because of the several reasons that I've explained earlier, because of those reasons, variety will eventually, variety will also deteriorate early. So the early deterioration can be prevented by these methods. Is that clear? So next we have to talk about seed multiplication ratio. So this is one of the important questions for your JRF and SRF exam. So make sure to just revise the seed multiplication ratio of different crops. Not only for JRF, they are also asking this for AFO exam and other agriculture exams. Wherever the agriculture is included, they directly ask some seed multiplication ratio of certain crops. So make sure to remember, make a list. Like I will include this in the IMACS table. Once I will include it, I'll tell you. So just revise them from time to time. And one more important thing to mention here is, there are a lot of sources from where you will get this SMR, but most of them are wrong. So many a times, like you see the different values for the same crop in the different source from the different sources. So the best way to find out the authentic SMR is to visit seed.net, from where I have just collected the SMR of all the crops for my SRF preparation. So anyhow, I am including it on this IMACS PDF. So if you want to check it from your end, you can visit that website and check it by yourself. So this is about SMR. So what is the formula to calculate the SMR? SMR is nothing but the seed multiplication ratio. So can anyone tell me, like, what is the formula to calculate that? So how do we calculate the seed multiplication ratio? So just remember these two formulas. So the seed multiplication ratio is nothing but it is equal to the seed produced divided by the total amount of the seed used, or the seed rate for a hectare. Like for a certain hectare, the number of seeds, like a total quantity of the seed that is produced divided by the seed rate that is used. So that will give us a seed multiplication ratio. So I have already also written it here so that you don't forget. Just refer this IMACS table PDF uploaded in the app. Okay. Next is seed multiplication ratio. Seed replacement ratio. So SRR. SRR is equal to X by Y into 100. So what is X and what is Y? Those X is nothing but the percentage of quantity of the quality seeds sown. So this is the quantity of the quality seeds that are sown this year, divided by, like, this year means the present year, divided by quantity of the seed that are required for entire area. For example, if we require like 100 quintals of a seed, if this season requires 100 quintals of the seed to be sown, but we are using only 50 quintals of quality seeds. Out of the total requirement, we are using only 50 quintals of the quality seed. Rest of the seeds, rest 50 will be from the previously sown, like farm safe seeds. The rest 50 will be from farm safe seeds. So what is the SRR here? So what will be the SRR? So is there any latency like from the time that you take to type and I see here? Because like every time like I see the responses, very delayed responses from you people. If I am using 50 quintal of quality seeds out of 100 quintals which are required, what will be the percentage? Yes, it's just 50%. So SRR is 50% here, right? I hope like now you have understood the formula. So there is a typical like this certain value where the hybrids, we know that since after F1 generation in the F2, none of them will be fertile since the use, because of the use of this male sterility system. So for the hybrids, the seed replacement is 100%. So you cannot use the farm saved seed here. Why the name replacement? Because we are replacing the existing or the farm saved seeds by the new seeds that are produced on that particular year. So what do you mean by the quality seeds? Quality seeds are nothing but they have came from the quality seed production, or just by the seed production that we are taking separately. It's they are not from the production or the cultivation of the crops for the edible purpose. See, earlier what used to happen when like we like as a farmers, they used to just grow the seeds for the consumption purpose, out of which they will just save certain amount of the seeds, then again they will resow in the field for the next season. So this is what was done earlier. So after knowing about or the knowing the importance or the recognizing the importance of quality seeds, the separate seed production system came into existence, right? So from which we produce the quality seeds. So now the meaning is we are replacing the cultivation by using the quality seeds that are produced by the seed production. So that is why it is a replacement. I hope like I have answered your question. Okay. So the for the hybrids, I hope like you people know why it is 100%, because we cannot save the seeds that are produced in this generation and use it for next generation. Someone is raising the hands. Wait. Okay. So that is why it is 100%. For the cross pollination, it is around 33, I guess. It is somewhere around 30 to 35. And for self pollinated, it is 25%. So this is again not that exact. No need to worry about it. So you just remember the range. That's it. For at least this is very exact for the hybrids. So make sure to remember it. So this was regarding seed replacement rate and seed multiplication rate. I hope like you won't be confused anything like any further from these two different topics, the SRR and SMR. Next, we have the system of mating. The system of mating is not from the breeding topic, which talks about the genetic assortative mating, disordertive mating. This is not the one. We are discussing about three different lines of systems, like one line system of seed production, two line system. No, never. Like they won't ask you like SRR of particular crops. Even if they ask, they can ask you about the cross pollinated or either self pollinated, or more commonly they will like they will ask for the hybrids. Like generally they will ask like what is the seed replacement rate for the hybrids. Since it is very exact, the value is very exact that it is 100%. Most of the time they will ask the hybrids only. Otherwise, like self pollinated, it is around 25, and for cross pollinated, it is around 30 to 35, somewhere around 33, I guess. So I was talking about the system of mating, which includes the one line system, two line system, and three line system. So these systems are related to seed production, or more specifically, hybrid seed production. So can you just decode it? What is one line system? What is two line? And what is three line? I will just give you the hint. ABR, and this is called as three line system. Since we have three different lines, so this is called as three line systems. And what about two line and what about one line? Whenever we require three different lines for the seed production, this is very common case of hybrid seed production. That particular system is called as three line system. So what about two line and what about one line? Why SRR is very less in case of self-pollinated than cross-pollinated? So what do you think? What will be the reason? It's just because of the pollination behavior, where the allelic frequency usually like is disturbed because of cross-pollination, right? Like the quantity of the seeds that you sow, because of the pollination, the genomic constitutions will be very diverse. So the yield that you are like the yield and other characters, it cannot be fixed in case of the cross pollinated or open pollinated varieties. But in case of self pollination, since from generation to generation the allelic constitutions do not disturb too much, so the seeds or the seeds that are used for edible purpose or the generation from which they are harvested, the same seeds, they since they have not deteriorated or they have not changed in their allelic combination or composition, so they can be again used in the next year. So that is why it's less, replacement is less. That's it. For example, what I mean to say is, like for example, you take a certain gene A is a gene which is directly correlating with the yield part or the yield of a crop, right? So now for example, a farmer is sowing a seed for this particular season, like in the June, let us take it as a rice. In the June or in the monsoon region season, like he will sow the rice seeds. So he will just sow the 50 quintal of the seeds and get around somewhere around 10,000 quintal of rice. So this is completely for edible purpose, right? So he has to market the seeds and earn money out of it. So what the farmer can do, out of this 10 quintal, again he can use 10,000 quintal, again he can just save that 50 quintal of this particular seeds and again use it for next generation. So why like there won't be any much change in the yield? Because since the constitution, allelic constitution is very much same, so again you will get a similar kind of like a similar quantity of the yield that he has received in the previous year. So there won't be much difference. But this is not true in case of cross pollination and often cross pollination or in case of hybrids. So that is why the rate eventually increases, like replacement rate eventually increases from self, cross, and hybrids. Is this clear, Asaswini? Two line system, GMS. How this is two line system? GMS is a three line system. Why? Because if you are using the genetic male sterility, you require fertility restorer gene. Without fertility restorer gene, you cannot produce the commercial seed. If the if from the any crop seed is of commercial importance, then you cannot produce it with a two line system. It is three line. Yes, it is EGMS. Very good. So it is either photoperiod sensitive male sterility or temperature sensitive male sterility. Okay, so this comes under two line system. So what is CHA then? CHA is nothing but use of chemically hybridizing agents. So this is very simple. See, whenever the certain farmer or certain seed producer want to develop a hybrid, what he will do? He will just use one parent as a female and one parent as male. If both are male fertile, he want to like he has to convert one into completely female line, right? So he will just sow one line and with the isolation he will just sow the another line. If it is A and this is B, so this particular lane, though there is no natural male sterility that is available, by the use of chemicals which are called as chemically hybridizing agent, he will make a complete plot into male sterile one, and the other one is male fertile. So he will transfer the pollens from the B line, and then you will get the hybrid, which are combination of A and B. So this is the use of CHA, where they are using only the two line system. One is used as a female, one is used as a female, which is converted into male sterile just by applying chemically hybridizing agents. So then the female, the pollens are collected from that particular plot and it is transferred to this female after the CHA application, and the hybrid seeds are harvested from the female line. Here, so that is why this is the two line system. So what are the other examples? Self incompatibility. How self incompatibility is a two line system? Whether it is a genetic or like a sporophytic or gametic. self incompatibility. What happens again, you have to use only two different plots or two different lines, where one line is completely self incompatible, so other one is normal. So if it is self incompatible, there won't be any self pollination that is taking place. It also always requires the pollen from the different parent. So that is how it can form, or it can give rise to the hybrid without the use of any sterility, like male sterility systems. So this is a two line system. So what about the one line? Like when we are using a clonal propagation, so that is again the one line system. We are not even crossing the two different parents. So if we are using tissue culture, this is again called as one line system. Is that clear? So three line, you already know it: CGMS, GMS, and CMS. Transgenic male sterility, is it one line? So can anyone answer this question, like whether the transgenic male sterility is one line system or two line or three line? Yes. So just Mausam, you should tell me now, if you have transferred a gene which is causing a male sterility in case of A. Let us assume the crop variety is A, like variety of a crop, name of a variety is A. You have transferred a gene to this and made it as a male sterile, right? So how do you maintain this male sterility? So you have around 50 seeds for this generation. So how do you maintain this particular transgenic male sterility to the next generation? A variety should be multiplied, right? So you cannot use all the A variety in cross with B and harvest the hybrid seed. Why? Because in the next generation you will be again short. You will have a shortage of A seeds. You will be not having A seeds. You cannot again and again transfer the gene. You just need to maintain it or multiply it from generation to generation. So how do you do it? You people are understanding my question? Just think about it. Utaya is answering is two line, three line. Nanika told it as two line earlier. Now it is three line. Normal line means, so we need to use another source for maintaining, or another line that should be used to maintain this sterility, right? So if we are using like any other like see fertility restorer genes can be also like maintainer in B line, which is which can be developed after producing a transgenic one. See the transgenic gene, if you are transferring it, and you can also produce the near isogenic lines of that particular one. So once the near isogenic lines is produced, you can use that near isogenic lines to maintain this particular A variety from generation to generation. Yeah, the examples is Barnes and Bursta system that is available in mustard. Yes, very good. I think I didn't include this mustard, Barnes and Bursta in this particular one. Is this covered in the plant breeding part? Did he cover the mechanism of Barnes and Bursta? Utay, you remember like whether Ari Vijay has covered this in this breeding or not? Okay, fine, good. So next, I was talking about lines of breeding. It is method. Next, we'll talk about the methods of hybrid seed production. So why we like? See, I will tell you like why this sterility system is very important for the hybrid. So just imagine a situation where you have two varieties. Let us assume it is Arun and Varun. Arun and Varun are the two varieties where the first variety is giving higher yield. Yield is good, but it has low adaptability to the environment or low disease resistance. but the second one has a low yield but higher disease resistance. I just simply taken only two different characters or two different traits for your understanding. So this is now 1 and 2 variety. So now breeder wants to just cross these two so that he will get a composition or effective composition where it will be higher yield with higher disease resistance. So how we can do it? How can you do it? In a large scale, I'm talking about, not only the single parent. In a large scale, if you want to develop a hybrid, how you should do it? So he has to sow the both the varieties in isolation separately. So either of these two, one has to be used as female and one has to be used as male, right? So if he is using this as a female, what about the pollens that are produced from this plant? For example, this is the variety of rice. In the rice, we know that it is self-pollinated. It's self-pollinated crop. Even soon after the flowering, there will be pollination. If the pollination is occurring, how can he transfer the pollens from Varun to Arun, like from that variety to this variety? If like soon after the flowering, within this variety of Arun, like self-pollination is taking place, and all the pollens or all the ovaries are fertilized now. So how to do it? So that is the reason we require some effective system which can convert these hermaphroditic flowers to completely male sterile one. So once it is completely converted into male sterile one, even if it like sows the hundreds of seeds, all the seeds will have only the ovary, exposed ovary or matured ovary. There won't be the pollen which is functional. So that it will be receptive. Whenever the foreign pollens will come on this and land on the stigmatic surface, it will pollinate and the seeds are formed. So for this to happen, it hermaphrodite should be converted into completely a male sterile line. It is not for the Arun like specific. It can be Varun, like any variety. So one variety should be compulsorily converted from hermaphrodite to a male sterile one. So how to do it? What are the different methods that can be used to convert this particular flowers into a male sterile one? So those are the male sterility. I'll just show you the things. Yeah. So there are 7 different techniques how you can do it. Either by the use of cytoplasmic genetic male sterility, genetic male sterility, or simply this is a male sterility. Next, we can use the self incompatibility, and next it is a pistillate condition. I will explain what is pistillate. And next we can use CHA, which I told you earlier, or if nothing is available, no source is available for male sterility, no source is available for self incompatibility, and there is no pistillate condition available, or no chemical is active, or no chemical is having any effect on that particular crop, ultimately we should go for manual emasculation and pollination. So these are the certain ways by which we can convert a certain variety into male sterile one, by which will help in the production of hybrid seeds. So I have just made a detailed note of the emasculation, and emasculation, what we commonly know is and emasculation, right? So in the other than the and emasculation, we have several other techniques that have the least importance, but just go through it once. So most common method is the and emasculation. We just pull the anthers and we'll just remove that. So that is the and emasculation technique. So apart from that, we have also like suction method, hot water emasculation, alcohol method, cold treatment, genetic emasculation. So these are also there. Just go through it once. They might be not that important, but at least you should know that there are certain other types or other methods of emasculation. Once it is emasculated, what is the next procedure? It is bagging. Why the bagging is required? Once you have emasculated the female, or you have emasculated a plant where the only the female part is receptive now, from to prevent the unwanted pollination on that, you need to bag it. So once you get a pollination, or once the pollens are produced from the variety that you require, you can just collect the pollen from that particular variety and just remove the bag and immediately dust those pollen on that one, on this particular variety, and this is how the seed set will occur. Okay, no need to worry about these types, but I think like you should go through it once. Okay, so what else is left? Pistillate lines. So what are the pistillate lines? So the pistillate lines are more commonly used in case of castor seed production, like hybrid seed production of castor, where the pistillate lines are differentiated into N type, S type, and NES type. I will explain you like what are this N type, S type, and NES in case of castor production. Once we talk the castor seed production, I will do it. So, but now let us understand what is this pistillate line. So, can anyone tell me like what are the pistillate lines? What do you understand by these pistillate lines? So pistillate lines are those, even though they are hermaphrodite, even though they have the ability to produce both the anthers and stigma, that particularly depends on the environmental conditions or the day length. For example, under certain temperature above 32, they are all male sterile, or they are completely pistillate because of the varying temperature condition. If the temperature is above the 32, they are all pistillate or male sterile. If the temperature decreases from 32 or below 32, they become male fertile. So there is a sex reversion that is occurring because of variation in the temperature, the surrounding temperature. So that is how this particular mechanisms where the male sterility system is reverting because of the environmental conditions. This is used as a case of hybrid seed production, and this is more commonly seen in the castor, where based on the temperature there is a reversion of the sex. So this particular mechanism is used for hybrid seed production. Pistillate is nothing but only forming the, yes, they are the all flowers are female. This is how it is converted. Like initially they are not only female lines, because of exposure to the certain temperature conditions, all will be pistillate in nature, and whatever the male lines are produced, like or male flowers are produced, they become sterile, or the pollens are non-functional. So this is how pistillate lines are used. So there are, like you might have heard about male sterility system, which you know more, like you are very familiar with genetic male sterility, cytoplasmic genetic male sterility, and like cytoplasmic male sterility, right? Three different systems. Do you know about any other male sterility system which are non-genetic in nature? One is EGMS, environmental sensitive male sterility. So what are the rest? So it might be because of nutrient deficiency, enzyme balance. If there is any disturbance in the enzyme balance, that will also again lead to the male sterility, or the ecologically induced male sterility because of the environment or the ecological behavior. There might be the expression of male sterility system or suppression of the male fertility. So these are nothing but the induced one. The male gametocytes can be chemicals. Then what about genetic male sterility? There are genetic male sterility that you have already known, which are you are very familiar with: GMS, CMS, and CGMS. There are other three types, which are functional male sterility, structural male sterility, and sporogenous male sterility. So I mentioned all these three things here that might not be that important, but as a student of plant science, and if you are going for plant breeding in your MSc, you should know it. Like what is structural male sterility, what is sporogenous, and what is functional, and what are the difference between these three types. And it is very simple to understand. Only thing is you should remember them. So this can be important. So just try to remember like environmental sensitive male sterility. So the TGMS, where like if the temperature is around 20, like more than 28 to 32, the fertility is lost, or the pollen will be not functional, which will lead to the male sterile line. But male fertility is restored if the temperature decreases to below 24 degrees Celsius. So reverse TGMS means like if less than 24 it is male sterile, and if more than 24 it is male fertile. So PGMS normally, when the day length is less or less than 14 hours, it is male sterile. If it is more than 14 hours day length, then it is male fertile. So this is just a common example. Okay. So what you should remember is environmental sensitive male sterility. Normally, higher temperature will lead to the male sterility. Lower temperature will give them restore the fertility. And secondly, when we talk of photoperiod, if the day length is more, then it is more fertile. Like if the day length is more, then it is male fertile. If the day length is less, then it is male sterile. So that is what you should remember. So this particular slide is of commercial importance, or it is important. Why? Because this is actually used in case of seed production. What I mean to say is for cereals. Like if someone asks you like how the hybrid seed production is carried out in case of cereals. So majority of the cereals we use the CGMS system where we have the effective male sterility system and fertility restorer gene. So can you name the male sterile cytoplasm for rice? What is the male sterile cytoplasm commonly used for rice? What about bajra, sorghum? I hope like you people have made the list of this cytoplasmic source, cytoplasmic male sterile source. Yes, it's wild about you. So you just, I hope like you are catching my points, right? Okay. So these are the things. Just try to remember. Try to make a list of all the male sterile sources for the major crops so that you won't forget it. But except for maize, where the CGMS is not like, even though we have certain CMS like cytoplasm lines or cytoplasmic lines, we don't use it. Why? Why? The reason is very simple. How many of you have seen the maize crop in the field? Just raise your hands. Like, have you seen the maize crop in the field? No one. Okay, some are raising your hands. Nice. Yes. Okay, four of you are rising. Okay, fine. So why the, like, even though like and emasculation is laborious when it is compared to other systems, and also it can lead to the errors. Why? Because it depends on the skill of a labor of how he is emasculating the crop. If he is not competent enough to emasculate it properly, there occurs the problem of quality. Like there occur the problems of genetic deterioration or the impurity or the presence of off types. So though there are disadvantages in the and emasculation, still in the maize we follow detasseling. What is the reason? The reason is very simple. It is very convenient and easy to remove the tassel from the maize field or the maize crop. It's very easy and convenient. We do not even need the Caesar to do it. Scissors, forceps, or nothing is required. You can just pull out the tassel from the maize plant directly without creating any damage to the crop. So that is why detasseling is commercially used in case of maize. That is the simple reason. So next, sunflower, we have CGMS, and it is again by the insect pollination, we know it. And the safflower, it is commercially like GMS is used, and but this is not used in India. And next, red gram is again like, you know that there are certain varieties that are been like hybrids that are being developed by the use of GMS system. Castor, I told you like the pistillate lines are like mostly used for commercial hybrid seed production. And next we have rapeseed and mustard, where we also have CGMS self incompatibility. Both systems are there, and it depends on the hybrid that we are going to develop, on which system we are using here. And lastly, the cotton. So we do not have an effective cytoplasm for cotton, so that is why you use the and emasculation and the pollination technique. Is that clear? Like I have just tried to mention few of the crops directly here, so that you will be able to remember like what are the different methods or mechanisms for hybrid seed production. So rest of the things, like for mostly for the vegetables, it is and emasculation and the pollination, or either self incompatibility. There are least chances that they use the CGMS in case of vegetables. For example, solanaceous crops, which are tomato and brinjal, it is mainly because by the process of and emasculation only. And for the coal crops, it is either and emasculation or self-incompatibility, and all other vegetables it is mostly anamasculination. So this is what you can remember. So now we'll talk about different schemes for seed production in case of India. So in India, I told you about the one important organization which is located in the MAU, which is Indian Institute of Seed Science. So what is the main function of this Indian Institute of Seed Science? It produces the indent, or it coordinates the production of breeder seeds in case of India through the AICRP, which is All India Coordinated Research Projects. So that is why this particular organization is very important, which coordinates the breeder seed production in India. It is Indian Institute of Seed Science. It is located in MAU. Just remember, MAU, you can expect this question in the exam, right? So this is a one institute that is involved in the seed production, like mainly like other institutes, like I already told you, like when it comes to certified seeds, even the progressive farmer is involved, cooperative societies are involved, private companies are involved, ICI institutes are anyhow involved from the foundation stage only, right? So all other institutes are involved when it comes to certified, but when it is from starting or beginning stage, it is from Indian Institute of Seed Science, where the breeder seed production is coordinated, right? So secondly, we have certain schemes to boost up this seed production. So one is farmers participatory seed production. So this is done to enhance the availability of the quality seeds. Since I told you, like the workforce in case of government, like for the government is very less, it cannot compensate or it cannot provide the quality seeds for all the farmers. That is why they are encouraging the farmers to participate in the seed production, which is by the scheme farmers participatory seed production. Okay. Next, what it will help for? It will help in the crop diversification, so that more farmers will take this seed production, and different varieties and different crops will have the quality seeds. If the quality seeds are available for all the crops, then definitely the yield will increase, so that the farmers who are seeing in terms of commercial basis, they will just buy these quality seeds and they will sow them. So the crop diversification occurs. So this is about farmers participatory seed breeding, so seed program. So next we have a concept called as seed village program or seed village concept, which is in Hindi called as Beej Gram Yojana. So what is seed village? So seed village is nothing but this is also an incentive or the encouragement that is given by the government to encourage or to involve the village in the seed production, wherein the trained group of farmers are involved in the production of the seeds of various crops. So this is how they will make available to the public the quality seeds. So what they are doing is they are just organizing the seed production in the clusters on the compact area, which will help in the replacing the local varieties and increasing seed production and the meeting the local demand. Why? Because if it is done in the village level, this circulation of these quality seeds will also takes place in a both formal and informal way. Informal way in the sense, like if the farmer is producing the seeds, he will definitely give those seeds, quality seeds, to his neighbors, right? So there is always a system of barter that is taking place in the village. So this is how there will be a spread of quality seeds, and this is how the seed village concept is helping in the distributing these quality seeds. Okay, so next is suitable areas. So when we talk of seed production, there are minimum criteria that are to be fulfilled or that are to be taken care of. So the area that are used for the seed production should be adapted to the photoperiod and the temperature conditions, and there should be a moderate rainfall and the humidity and a moderate temperature and gentle winds in case of which is very much necessary for the wind pollinated one, and thus there should be ample sunshine. So it generally means that all the conditions should be of optimum, or in the ambient conditions should be there for the seed production to take place. It should be like a cold and dry weather, cool and dry place, not a heavy rainfall area or not the high temperature areas. It should be like everything should be in a medium level. So this is the place where you can take the seed production. So what about India? Like if someone asks you, so tell me some of the important areas where the seed production is taking place, majorly taking place. So this comes to the Averi. This is in Karnataka, Karimnagar, Aurangabad, and also some parts of Gujarat. So these four areas are mainly for field crops, and for coal crops it is in Kulu Manali. And for vegetables, this is the hub, seed hub, which is Averi, which is Ranebennur, which is a seed hub for the horticulture crops. And nextly, it is Aurangabad. So why this is very specific to this region? Because of the moderate rainfall, low cloudiness, presence of the compact area which is necessary for the field inspection. See, if the area is very small, like land holdings are very small, so it is very difficult for the field inspection to take place, right? Because I told you, like the unit, like they will be distributed as a unit. So unit will have the field inspections by the seed certification officer. So all these things will be difficult if the area is too small. So the compact area will help in the effective seed production and presence of higher land holdings, and the compact area, a fixed area. Okay. Mm, yeah, it's already of 1 hour from now. Okay, no problem. We can discuss this IMSA standards and also the seed production in the different crops in the next class. Is this clear? Like whatever I have discussed, is this clear? Is that clear to you all? Any doubts from today's class? Or you want me to go a little bit slow, or it is fine? You want me to go a little bit slow, you can just tell me. I need some answer, like yes or no. Like yes for what? Like you want me to go a bit slow, or yes for like you have understood everything? Okay, fine, thank you. So we'll meet in the next class where we'll start our discussion by IMSA standards. So this is very important, where you might expect some direct questions. So make sure to attend it and take a full benefit out of it. Yeah, I will upload it today. Probable exam date. I discussed this in the beginning of today's session, that you might expect this JRF exams within the end of this July. You can expect it from today. You can expect it from 45 days. 45 to 60 days, maximum of 60 days and 45 days, like either in the second or third week of July or last week of July. Like this is based on like the timing that every year the application is released. So based on that, I'm saying it. It's not any inside news that I have received from anyone. It is only based on the previous years, like where whenever the application is released, they will give a 15 days of time to announce the date, and after that they will again give the 15 days of time for the exam date. Okay, exam to be conducted. Okay, so if things are clear, we'll meet in the next class. Thanks for attending.